Good morning, everyone. Thank you for joining us. Welcome, Chantell. I have the pleasure of introducing you before you start your talk. Do you want to share your screen?
Yes.
Okay.
I can do that.
Chantell Evans is an assistant professor of cell biology at Duke University. She received her PhD in molecular and cellular pharmacology from the University of Wisconsin and was a Howard Hughes Medical Institute Hannah Gray postdoctoral fellow at the University of Pennsylvania. Her lab uses advanced microscopy and biochemical techniques to gain insight into the various mitochondrial quality control pathways in neurons and how mitochondrial regulation contributes to neurodegenerative diseases such as Parkinson's disease and ALS. Chantell is the inaugural recipient of the Science Diversity Leader Award from the Chan Zuckerberg Initiative and a Freeman Hrabowski Scholar recently named from the Howard Hughes Medical Institute. Congratulations and thank you.
All right. Well, thank you so much for that kind introduction. And yes, very excited about the new funding through HHMI. And really excited to be here today to share with you guys some of the work that I did as a postdoc and now as our lab is kind of getting up and running. We're really focused on understanding mitochondrial quality control and how that contributes to neurodegeneration. And so with that, I will go ahead and get started. Let me get a laser pointer so you can see. All right. All right, so in our lab, we spend a lot of time thinking about neurons and how they work. And I love to start out by showing the textbook drawing of a neuron. So here you can see the cell body or the soma, which contains the nucleus. You see these short dendrites that are coming off of that soma. And then you see a slightly longer axon. And through that axon, we know that a lot of organelle communication, and this is what allows for propagation for an axon to be then released synaptic vesicles at this distal tip to allow neurons to communicate with one another. However, this textbook drawing does a terrible job of showing you the complexity of these cells. However, when we show them drawn to scale, I think this puts a much better representation. So here's a motor neuron drawn to scale. This tiny red dot, again, is representing this soma or cell body. And then this long continuous line winding back and forth is the axon. And so I think from this perspective, it does a good job of showing the complexity of these structures and how important it is to know when and where a protein or organelle is within this axon, and how if you have defects in these maintenance mechanisms, how over time they can start leading to degeneration. And so when you take this and think about what's happening within the brain, there are about 86 billion neurons within the brain. And so their ability to form these complex structures are very important. And so we know that neuronal homeostasis can be maintained by the autophagy pathway, which helps promote axonal health. And this allows for the removal of cellular waste. And so within this pathway, you have the formation of this phagophore, and what it starts to do is just form and start to engulf cellular debris. Now, when I say cellular debris, this typically is damaged proteins, oxidized lipids, or small bits of organelles such as mitochondria. Once this autophagosome then forms, it then fuses with a lysosome, generating this autolysosome, and then you can get the breakdown of this cellular debris and recycling of these components. Now, this is the basal autophagy pathway, and this is, again, is a non-specific bulk engulfment. So whatever debris is out there within the cytoplasm is going to then engulf. And so we know that this autophagy pathway is important for maintaining cellular homeostasis, and that when this pathway is dysregulated, it can lead to degeneration. And this is important because, again, for within that axon, there's a lot of transport that's happening where you're having supply and clearance of proteins and organelles, and how important that is for, again, the spatiotemporal signaling for those neurons to be able to communicate with one another. And we also know that neurons rely heavily on mitochondria to support local energy demands at those fusion sites that are located far from the soma. So the mitochondria, I always like to start out by saying, is more than just the powerhouse of the cell. The mitochondria is a very unique organelle in that it is double membrane. So it contains an outer membrane as well as an inner membrane. And within this inner membrane are where all of the components and proteins for the electron transport chain are to generate ATP. Mitochondria are also unique in that they contain their own DNA, and that the DNA that is contained there are synthesizing these proteins that are needed for generating the electron transport change. So in addition to generating ATP, we know that mitochondria are important for metabolism. They can trigger cell death. They do a lot of calcium storage. They can do some cell signaling. And now more recently, we found out they're involved in innate immunity. And so here is a 3D rendering of a mitochondrion. So shown here in red is the outer mitochondrial membrane. In yellow is the inner mitochondrial membrane. And you can see that those two membranes are actually very close together, and they're only separated about 20 nanometers. And then you have the cristae, which you can see is taking up actually most of the structure of this organelle. And again, this is really important because that's where all of those proteins are generated in order to make ATP. And so in thinking about how these mitochondria are located within a neuron, showing you that here, so our mitochondria are shown in blue, and the membrane is marked by this yellow brownish color. And so within the neuron, you can see if we look in the soma or cell body, there's a dense population of mitochondria that are here. And then if you look out within the axon, within all of these structures, you can see that the mitochondria just kind of sprinkled throughout all the way even to these distal tips. And what's really interesting is that we know that there are about 2 million mitochondria per neuron. And if you do the math, so the fact that there are 86 billion neurons within the brain, this ends up being about 172 quadrillion mitochondria, or 1.72 times 10 to the 17th. And what I'm showing you here is an electron micrograph. And again, it looks very similar to what you the drawings where you can see these nice cristae that are forming, where all of those energy has been being generated via ATP. You can see that there's some mitochondrial DNA within there. And so what the question we're really interested in understanding is, how are mitochondria mechanistically regulated to help maintain neuronal homeostasis? And so in addition to this basal autophagy pathway, there's a selective form of autophagy. And within this pathway, it allows cells to specifically eliminate specific organelles. And so we focus on this mitophagy pathway, which allows for the cell to get rid of damaged or aged mitochondria. So within this pathway, there are actually multiple flavors that exist, but today I'll be highlighting this Parkin-mediated mitophagy. And so what ends up happening is you have some form of oxidative stress that triggers damage to this mitochondria. In our cell culture model systems, we can mimic this by treating our cells with reagents that damage the mitochondria, such as CCCP or antimycin A. But once you have damage to this organelle, this leads to the recruitment and stabilization of PINK1 on the mitochondrial surface. This then recruits an E3 ubiquitin ligase Parkin, and together these two proteins will start to phosphoubiquitinate outer mitochondrial membrane proteins. I'm only showing you two here, but you can imagine that this is a pretty heavy feed-forward mechanism, so you're going to have a lot of ubiquitin chains that are coming off of this organelle. And if there was a healthy mitochondria right next to it, it would not have these ubiquitin chains. So this triggers signals to the cell as an eat me signal, saying something's wrong with this particular organelle and that it needs to get degraded. And so this leads to the recruitment of these autophagy receptors, which I'll talk about within the next slide. And then it gets engulfed into an autophagosome. And then through lysosomal degradation, then the lysosome comes and fuses and releasing those enzymes. So now we're getting specific degradation of just that damaged mitochondria. And so one of the major players within this that I'll highlight today is this protein called optineurin, which we know is an autophagy receptor for this particular mitophagy pathway. What we mean by that is that it gets recruited and helps to trigger this autophagosome sequestration. So there are two particular domains within the protein that are particularly important. One is this LIR domain, which allows for optineurin to bind to LC3 on that growing autophagosome. And the other is this UBANE, or ubiquitin binding domain, which allows for optineurin to bind to ubiquitin. So you can think of it kind of as a bridge, bringing these two structures together in order for the autophagosome to sequester that damaged organelle. And what I'm showing you here is what it looks like via microscopy. So you can see this optoneurin shown here in yellow, and it's forming this ring, and it's engulfing this mitochondrial fragment shown here in magenta. And so we know that optoneurin is important for this mitophagy pathway, but it also is an autophagy receptor for the removal of salmonella through xenophagy. We also know that it can act as an autophagy receptor for the removal of protein aggregates, which is agrophagy. And so in addition to knowing a lot about this optoneurin protein, we also know that it can be stabilized on the mitochondrial surface through this kinase TBK1. So we can visualize optoneurin recruitment to damage mitochondria in these non-neuronal cell lines like HeLa cells that I'm showing you here. And so global treatment with CCCP induces optoneurin recruitment, and so does a more limited form of mitochondrial damage shown here using this optogenetic approach. So these cells are expressing an untagged Parkin, and I'll note that recruitment is dependent on Parkin overexpression because these cells don't express high endogenous levels of Parkin. And then optoneurin shown in green, and then this mitokiller red, where light-induced activation of mitokiller red causes local damage via ROS production. So in this video, you'll see light activation within these yellow boxes, followed by optoneurin recruitment. So over time, you will start to see optoneurin becoming much more bright. And you can see that optoneurin is only being recruited in the places where we showed light, so within those yellow boxes. And then you can see it starts to form these rings around these damaged mitochondrial fragments. And shown here is just a line scan highlighting that. So we have one peak for our fragmented mitochondria, and then two peaks in green showing that optoneurin is indeed engulfing that in a single frame. So looking downstream of optoneurin recruitment, we can also visualize the translocation of LC3, which again is a marker for that autophagosome. So 90 minutes after CCCP treatment in these HeLa cells that are overexpressing Parkin, we see recruitment of optoneurin as well as LC3 to these damaged mitochondrial fragments. And again, these line scans are highlighting that both optoneurin and LC3 are around the mitochondrial fragment in this single Z plane. So to confirm that optoneurin initiates autophagosome formation, we knocked down the protein and looked at LC3 recruitment. So in mock conditions, we see that LC3, it translocates to the damaged mitochondrial fragment following CCCP. But if we knock down optoneurin, we no longer see LC3 recruitment, highlighting that LC3 is downstream of optoneurin. Additionally, we express an optoneurin mutant that lacks LC3 binding activity. This is this optoneurin F178, and observed loss of LC3 recruitment to the damaged mitochondria following CCCP treatment compared to our wild type conditions. And this is quantified here on the right, where there's a significant decrease in the percent of mitochondria that are positive for LC3 in cells expressing this optoneurin mutant compared to cells that are expressing wild type optoneurin. And so in addition to knowing quite a bit about this mitophagy pathway, we know that mitochondrial dysfunction is also implicated in multiple neurodegenerative diseases. And so to highlight that, I'm showing examples of the mitochondria in rat brain. So this is young, so only 10-month-old, and then old would be 22 months old. So in the young, here we're highlighting the mitochondria with these red arrows. You can see a nice dense structure. They're smaller. That dense structure is coming from all of the cristae that are contained within it. And then if you compare that to the old, one of the first things you notice is that that dense structure is no longer there. So we've lost a lot of the cristae structure contained within those mitochondria. And then also they're quite swollen and large. And so we see that there's increased mitochondrial stress and dysfunction with age alone. And this has led to mitochondrial dysfunction being implicated in multiple neurodegenerative diseases, including Parkinson's disease, Alzheimer's, Huntington's, and ALS. But in addition to just these implications, we also know that there are genetic links to these diseases. So in this mitophagy pathway, again, this Parkin and pink-1 protein that I highlighted within the introduction, we know that those are found in both familial and sporadic cases of Parkinson's disease, as well as TBK1 and optineurin are known to be involved in both familial and sporadic cases of ALS. And so currently these diseases have poor early stage diagnosis, symptomatic treatment options, and no known cure. And so neurodegenerative diseases are continuing to become a major threat to human health. So finding biomarkers, treatments, or cures carries quite a bit of increasing urgency, because the World Health Organization has predicted by 2040 that neurodegenerative diseases will overtake cancer as the second leading cause of death worldwide. And so our lab and others really think that having a basic mechanistic understanding of how these pathways work in order to maintain mitochondrial health will be crucial in helping to come up with some of these biomarkers and treatment. And so what's interesting and nice that we can do within our cell system is that we can take those disease-associated mutations and then put them into our cell line and say, what happens to this pathway as a result of that? And so here I'm showing you a Parkinson's disease-associated mutant of Parkin that lacks E3 ubiquitin ligase activity. So in wild-type conditions, after treatment with CCCP, you see that Parkin nicely forms these rings and gets recruited to the mitochondrial fragment and co-localizes with optineurin. However, if you overexpress a mutant form of Parkin, while it still gets recruited, it has no E3 ubiquitin ligase activity, and so it does not put those phosphoubiquitin chains out, and so optineurin cannot recognize that. And so it stalls this mitophagy pathway. And if we look at longer time points, so instead of 1 hour, we look at 3 hours, we still see no optineurin recruitment. This is an ALS-associated mutation of optineurin that lacks ubiquitin binding activity. Again, in wild-type conditions, optineurin gets recruited and forms those rings around the fragmented mitochondria that co-localizes with LC3, the marker of the autophagosome. However, in a mutant background, optineurin doesn't get recruited to the mitochondria because it lacks ubiquitin binding activity, and therefore we don't see LC3 recruitment. And finally, this is an ALS-associated mutation of TBK1 that lacks optineurin binding activity, and very similar to the other two, in that mutant background, this protein doesn't get recruited. And so we see that expression of these disease-linked mutations either slows or disrupts efficient mitophagy, which leads to the buildup of damaged mitochondria that could ultimately lead to cell death. And so this suggested to us that mitochondrial turnover may be a key mechanism that's contributing to neurodegenerative diseases. And so we were really interested in how is this pathway happening in neurons? And so when I was in my postdoc and started doing a lot of literature searches to understand what was known about mitophagy in neurons, I was surprised to find that it was a pretty limited. And due to the unique characteristics of neurons, there are several basic questions that are really understood, such as, are distal and somal mitochondria subject to the same quality control mechanisms? If you're a mitochondria that's located in the soma or one that's located in the axon, are you regulated the same? Are you regulated differently just based off of your position within the cell? Second, where does mitophagy occur within these highly polarized cells? Do we see it happening everywhere, or is it limited to a particular compartment? And then finally, how quickly are these damaged mitochondria being removed from the cell? You can imagine in a cell that needs to be maintained for a lifetime, that removal of these damaged organelles would be particularly important. And so with this knowledge of the various pathway components in time course, we asked how molecularly and spatially conserved is this pathway in neurons? We know that from the time course of mitochondrial damage, it takes about 40 minutes to get through autophagosome engulfment, and this is within a non-neuronal HeLa cell line. So what could this look like within a neuron? And so in order to test this, we used primary hippocampal neurons with endogenous Parkin levels, and then we induced mitochondrial damage via global oxidative stress through antioxidant removal, or we used a positive control as a low dose of antimycin A. And I'll note that we use this mild oxidative stress to mimic highly metabolic cells. And so with that, we could then ask, what are the spatial and temporal dynamics of these various mitophagy-associated proteins to the damaged organelles? And that we could also, by using and relying heavily on live cell imaging, that we could look at this within different cellular compartments. So to establish our mitochondrial damaging paradigm, we looked at what effect our treatments had on the mitochondrial network. And so what I'm showing you here are 3D renderings of the somal mitochondrial network in both control and our antioxidant-free treated conditions. And we see a nice elongated mitochondrial network in both control and treated conditions, which is indicative of a healthy mitochondrial network, suggesting that our damaging paradigm does not significantly alter the mitochondrial network. We then could also look at mitochondrial function. And by that, one of the things we used is TMRE. So this is a dye that labels healthy mitochondria. So in control or antioxidant-free conditions following 6 hours, we saw no global difference in the mitochondrial health or membrane potential with this TMRE. But then we looked at cell ROX, which is a marker of intracellular reactive oxygen species. And we saw after 6 hours in our antioxidant-free condition, there was a significant increase in the cellular ROS compared to control conditions. And then we did this at 7 DIV. We also repeated these experiments at 14 DIV and found very similar results. And so I also will note that we looked in the axon. Everything I'm showing you here is soma. We also looked within the axon. We saw no differences. So our global antioxidant-free stress paradigm is not affecting one population of the mitochondria compared to the other within a cell. And so again, we see that there are no global changes to the mitochondrial network, despite these increases in intracellular ROS. Again, suggesting that we're inducing very low levels of damage without compromising the somal network, the cell viability, which we think is really important and is much more physiologically relevant to what could be happening within these neurons over years of time in order to lead to the development of neurodegeneration. And so in neurons, we can visualize optineurin recruitment to mitochondria following an hour of antioxidant removal. So what I'm going to show you within these videos are a hippocampal neuron, the soma of it, that's expressing this optineurin protein shown in yellow. We have a mitochondrial marker that's shown in magenta. And then we're doing confocal imaging where we're taking Z stacks and through different Z planes in order to look at the mitochondrial network. So within this video, you can start to see over time, not over time, but through the different Z stacks that we see, optineurin rings that are forming. Again, they look very similar to what we saw within those non-neuronal HeLa cells. So if we take one single Z plane, shown here, again, you see that optineurin ring, and if you look within it, you find that fragmented mitochondria. And so we see these events that are occurring throughout the soma and located through these different Z depths. So with that, what we can then do is use Amaris software to generate 3D reconstructions of what those cells look like. So that's what I'm showing you here. That was done by Yuli Martinez Castillo in our lab. So this is, again, the mitochondrial network within a single cell. These mitochondria are shown in magenta. We're highlighting the fact that we can see these optineurin rings. And if you look within that optineurin ring, you see that fragmented mitochondrial marker. Again, the mitochondrial network looks intact. The cells are very healthy, but we're still able to induce this low levels of mitophagy using these antioxidant removal. Okay, so we determined that optineurin is being recruited to these mitochondria, but then we had to say, is this specific for damaged organelles? And so again, to do this, we turned back to this TMRE marker, which labels healthy mitochondria. So if you look, the TMRE marker tracks very nicely with this transfected mitochondrial marker for these nice elongated mitochondria, again, which are markers for healthy mitochondria. But then what we also were able to routinely find is our optineurin rings. And if we look within our optineurin rings, we find the fragmented mitochondrial marker. However, these are negative for TMRE. And so this is telling us that optineurin translocation is specific to damaged mitochondria, and that we can use this to positively identify these mitophagy events shown here on the right-hand side. And so then we quantified the percent of cells with optineurin positive mitochondria. We observed a significant increase following this antioxidant removal compared to our controls. This is at 1 hour. We also did this at 6 hours, and again, see very similar results where we find a significant increase following antioxidant removal compared to control. And this is also just showing our positive control with that low dose of antimycin A. Okay, so I previously mentioned that upon damage, we get pink 1 and Parkin recruitment to the outer mitochondrial membrane. And this leads to phospho-ubiquitination of these outer mitochondrial membrane proteins that leads to optineurin and TBK1 translocation. And so we transfected cells with these upstream mitophagy-associated proteins to determine whether we could also see these being recruited to our damaged mitochondria. And so what I'm highlighting here is that you can see that Parkin co-localizes very nicely with our optineurin, and you can see that these are both engulfing that fragmented mitochondria. And so in addition to looking at just Parkin recruitment, we also wanted to confirm that what we're studying is indeed Parkin-mediated mitophagy. So we used siRNA to knock down Parkin. We see a dramatic decrease in the percent of cells with these optineurin positive mitochondrial rings. However, if we overexpress or put back wild type Parkin, we're able to rescue that. And then we also saw a partial rescue with a Parkin mutation. So we established a novel mitophagy paradigm where this antioxidant removal induces low levels of mitochondrial damage without compromising the health of the mitochondrial network. And then the optineurin translocation and ring formation to these mitochondrial fragments is a positive identifier of these mitophagic events. And so with that, we were able to begin to ask our questions of interest. And one of them that we really wanted to understand are what are the temporal dynamics of neuronal mitophagy? So we know that this is a constitutively active pathway. So in healthy neurons, this pathway is turning over any damaged or aged mitochondria that need to happen. Where if you have cells that are expressing any of these neurodegenerative disease associated mutations, that this is going to stall this mitophagy pathway. And then this stalling will lead to the buildup of these damaged organelles. And so we hypothesize that failure to remove these damaged organelles would be detrimental to neuronal health. And that also, again, because of the fact that you have the neurons that you need to maintain for life, we thought the time course for the removal of these organelles would be particularly important. And so what we ended up doing is damaging our mitochondria through this antioxidant deprivation for 1 hour, and then asking how quickly do we see these various mitophagy-associated proteins being recruited to the organelle. And so within an hour, again, we were able to see the recruitment of Parkin. We were able to see the recruitment of ubiquitin, again, ubiquitin chains on those damaged mitochondria that allow for optineurin to bind. And so we see that they co-localize very nicely with optineurin. We also see the marker TBK1. This is, again, a kinase that phosphorylates optineurin and helps to stabilize on the mitochondrial membrane. And then finally, we see the marker LC3 for the autophagosome. And so I'll note that the translocation of these proteins occurred within the predicted time frame from what was previously characterized within those non-neuronal HeLa cells. And so we then looked at the final stage, which is lysosome fusion and acidification to fully degrade those damaged organelles. So in order to do this, we use the marker LAMP1, which is a lysosomal associated membrane protein. And this particular construct is hooked up to an SEP, a superecliptic fluorine. Think of it as very similar to a GFP, and then as well as an RFP. And by having these two probes together, it allows us to simultaneously visualize lysosome fusion and acidification. So in a non-acidified organelle, we'll see both the SEP and the RFP signal. But in an acidified organelle, this SEP signal will be quenched, and so we'll only be able to visualize the RFP. So we again identified optineurin positive mitochondria. We have found events that were LAMP1 negative, where the lysosomal fusion had yet to occur. We found events that were dual labeled with both the SEP and the RFP, indicating a non-acidified organelle. And we found events that were RFP only, suggesting a degradative competent lysosome. And so then we just scored our events for either being negative, dual, or LAMP1 RFP only. And what we found is that most of our events in either our control or 6 hour, either our control or antioxidant free, or looking at our 1 hour compared to 6 hour, we found that most of them were sitting in this dual labeled LAMP1 organelle, indicating that, again, it was non-acidified. And so to really understand and dig a little deeper into this data, what we had to do is kind of go back and revisit the path to lysosomal degradation. What I'm showing you is that we have been using this marker LAMP1, again, which marks the lysosome, and we can see it gets recruited and is very nicely localized to these optineurin positive mitochondria. But I just want to remind everyone of what it looks like for autophagosome sequestration to remove this cellular debris. So once this autophagosome fuses, it then fuses with a lysosome, and then this generates an autolysosome. And degradation in the lysosome only occurs through the enzymes that break down that cellular debris. And some of these enzymes are called cathepsins. So in an unacidified environment, these cathepsins are in a procathepsin state. And then under low pH or an acidified environment, they're cleaved into active cathepsins that allow them to break down the cellular debris. And so in using LAMP1 as a marker, we find that it's on late endosomes, as well as degradative competent lysosomes. But only degradative competent lysosomes contain active cathepsins. And so if we go back and revisit our data, when we're showing this dual labeled LAMP1 state, because it marks late endosomes as well as degradative lysosomes, we can't know for sure which one of these states or compartments our structures are marking. So this dual label state is actually a summation of these late endosomes that are degradative incompetent, as well as these degradative competent lysosomes. And so in order to go back and revisit this a bit more, what we ended up doing is rerunning these experiments, but instead of using LAMP1, we're now using cathepsin D, again, a marker for those cathepsins. Again, this is hooked up to an SEP and an RFP. So very similar, this construct works in the similar way to the LAMP1. So in a non-acidified state, you're gonna have the SEP and the RFP, where in an acidified organelle, you're only gonna have this RFP. We then went through and found optineurin positive events. And just like before, we found events that were cathepsin D negative, events that were dual labeled with both the SEP and the RFP, and finally events that were labeled only with RFP. We then scored our events again, and this time you can see that, that many of the events within the 1 hour in either control or antioxidant free are in a cathepsin D negative state. And then when we move to 6 hours, we start seeing more of them shifting to this cathepsin D RFP. And so this is pretty consistent with our previous findings where we observed that most mitophagic events in either basal or induced conditions are sitting in this, sequestered in this non-acidified organelle. In order to test this a little bit more, we also performed some Western blots to look at that. So what Jordan has done here is that she's treated her cells with different conditions, and at the same time has applied cyclohexamide, which stops protein synthesis. And then we could look at the turnover of our mitophagy-associated proteins, either Parkin, optineurin, cytochrome C to look at the mitochondria, as well as LC3 to look at our autophagosomes. And we can see in either our control or our various conditions going anywhere from 1 hour of antioxidant deprivation all the way out to 24 hours, that we're really not seeing any differences in these proteins, suggesting again that this lysosomal degradation isn't occurring as rapidly as we would think it is. And so from this, we came up with a central hypothesis that delayed degradation is intrinsic to neurons, making neurons more susceptible to stress and degeneration. And what we really want to be able to do is identify the temporal dynamics of this neuronal mitophagy pathway. And so one of the first questions we were interested in looking at is, what is the time course of mitochondrial turnover? And so in order to do this, we're using these age-related pulse chase experiments, where we're able to investigate long-term mitochondrial sequestration and elimination. And so to do this, what we do is we transfect our cells with a mitochondrial targeted SNAP tag. And this SNAP tag can covalently bind to SNAP ligands. And so we transfect this in, and then we come back the following day, we induce our mitophagy by damaging the mitochondria, and we do our first SNAP pulse. At this time of doing SNAP pulse, because our mitochondria also are entering the mitophagy pathway, they're going to be labeled this magenta color, and we'll be able to track them over time. We then end our mitophagy treatment, and then we come back on and put on a SNAP block. This SNAP block can rebind to any of the remaining SNAP binding the SNAP tag binding sites that we missed with our first pulse, but this is dark, so we won't be able to see this with our fluorescence imaging. We then allow our mitochondria to recover overnight, and then the following morning, we come back and we pulse in a second SNAP, which was shown here in green. And so what this ends up showing is that we have an old mitochondrial network that's shown here in magenta, a young mitochondrial network that's shown here in green, and then our network will be dual labeled because the mitochondria are undergoing constant fusion and fission events. But what we're really interested in seeing is that, can we identify these old magenta only mitochondria that that have entered this mitophagy pathway, and will they be LAMP1 positive or LAMP1 negative? And I'll highlight here that we had a paper that was published that goes over this protocol to do this pulse chase labeling. And I think it could work really nicely, not only for mitochondria, but for other organelles such as the endoplasmic reticulum, to where you can kind of look at the turnover or watch the processes of these organelles over long term. And so 24 hours after our initial damage, we observed optineurin positive mitochondria, shown here, and found that these mitophagic events were positive for our old mitochondrial marker. However, they were negative for our young mitochondrial marker. And so this indicates that these mitochondria have been sequestered for an extended period of time. What's even more interesting is that we see that these events also had not undergone lysosomal fusion, shown here by the absence of this LAMP1 signal. So again, this is 24 hours after we've induced our initial damage. We've now gone back and done experiments where we're now looking 72 hours after our initial damage. Again, we can find these optineurin positive mitochondria that are labeled with the old mitochondrial marker. They're negative for this young mitochondrial marker. And shown here, we see that this is an example of an acidified event, shown here by the positive lysotracker signal, which labels acidified organelles. So again, if we go through and we quantify whether or not our events that are optineurin positive are either positive or negative for lysotracker, after 24 hours, we see that most of our events are lysotracker negative in either treated or controlled conditions. And we start to see, again, after 72 hours, that now most of our events have pivoted and become lysotracker positive, showing that they are being acidified. It just takes an extended period of time. So again, we have this efficient sequestration of these damaged mitochondria into an autophagosome, but there's this slow, inefficient degradation to remove these mitochondria, where it occurs what it looks like to be almost days after the initial mitochondrial insult. So then the second question that we were really interested in following up on is, what is the molecular mechanism of delayed mitochondrial turnover? And we hypothesized that this could be one of two events. So first, it could be that our autophagosome fails to fuse with a degradative competent lysosome. Something's going on with its ability to fuse, and therefore we're not getting the fusion event to happen, and that's why we're not seeing turnover. However, a second hypothesis could be that our autophagosome fuses perfectly fine. However, it's fusing with degradative incompetent lysosomes. And so to test this, we've started doing some experiments looking at our autophagosome maturation, the lysosomal fusion, and as well as some of the protease activity that are contained within these lysosomes. So one thing that we've started doing is looking at local levels of ATP, because we know that ATP is important in order for those ions to get pumped into the lysosome in order to make it an acidified lysosome. And so to do this, Molly Gooden, a research technician within the lab, has started running these experiments using this A-team, which is a ATP sensor. So we can look at the fluorescence signal of A-team in its ATP-free state, as well as its ATP-bound state. And we've done this and quantified the A-team relative fret efficiency for our total, just looking within the neuron in either our control or our antioxidant-free conditions. And we can see at either 1 hour or 6 hours, there's really no difference in the overall levels of ATP within the neuron. We then went back and said, okay, if we don't see changes in the overall neuron, can we see changes locally around those mitophagy events? So then we went through, we identified our optineurin positive mitochondria, just highlighting an event here, and then looked, what does the local level of ATP look like around this mitophagy event? We then went back and again quantified the relative fret efficiency for just our mitophagy events in either control or antioxidant-free conditions for either 1 hour or 6 hours, and we saw no differences between the two. So we don't think that there are changes in ATP levels that could be affecting the ability of these lysosomes to be acidified to remove our damaged mitochondria. So another thing that we decided to look at to see, is this delay specific to neurons, or is it something that's a general that's happening within all cell types? So we went back and we turned to these non-neuronal HeLa cells because they're used so routinely within the mitophagy field. So we looked at lysosomal fusion, again, using this LAMP1 probe. We damaged our mitochondria and then looked at how quickly we found LAMP1 positive, optineurin positive mitochondria. So mitophagy events that were positive for LAMP1. We damaged for 90 minutes. So our zero time point is that 90 minutes. And we found about 50% of our events were already positive for LAMP1 after 90 minutes. We saw that generally rose to almost 100% after 6 hours. We then repeated these experiments looking at lysosome acidification using that lyso tracker and saw very similar trends where over time it pretty much maxes out at 100%. So we see that this delayed turnover may be specific to neurons. And so currently we're testing more neuronal and non-neuronal cell lines to really understand this. And so collectively, our data support a model where slow acidification could serve as a point of vulnerability, where factors such as increased levels of oxidative damage to organelles or changes in lysosomal acidification, which we know happens with age, or expression of mutations in pathway components could exacerbate this pathology. And so we hypothesized that alterations to the system might impede the rate of turnover, increasing neuronal sensitivity to damage leading to neurodegeneration. And so to test this idea, we expressed an ALS-linked mutations of both optineurin and TBK1 and looked at its effects on mitochondrial health and neuronal viability. And so again, we turn back to this mutation that I showed in the introduction, this optineurin E478G mutation that fails to translocate to damaged mitochondria and initiate mitophagy. We also looked at this TBK1 mutation M559R, where this mutation fails to dimerize, and that affects its ability to translocate to damaged mitochondria to mediate this mitophagy pathway. So again, here in this mutant form, you see this protein does not get recruited to the damaged mitochondria, even though Parkin gets recruited. And so we expressed both of these mutations to really understand what was happening. And so what I'm showing you here is we turned back to this TMRE to measure the mitochondrial membrane potential, or health. So just to highlight first, to pay attention, if you compare just cells that are expressing wild type optineurin to cells that are expressing the mutant form in either control conditions, you can see that mutant expression already decreases network health. We then see that if you add oxidative stress on top of mutant expression, that this further exacerbates the effect, which we think is enhancing the susceptibility of the neuron to neuronal stress. So not only do we see this with this optineurin mutant, we also see it with this TBK1 mutant. Again, mutant expression alters mitochondrial health, and then mutant expression with oxidative damage exacerbates this effect. And so to conclude, we've characterized this novel mitophagy induction paradigm where antioxidant removal induces low levels of mitochondrial damage. And finally, we looked at the temporal dynamics and observed that there are these that there's slow mitochondrial turnover in neurons that is occurring, hours to days even after initial damage, and that we think that these slow kinetics may enhance susceptibility to these disease-linked mutations and lead to neurodegeneration. So as I've mentioned or hinted at kind of within the introduction, that we know that a hallmark of many of these neurodegenerative diseases is this accumulation of dysfunctional mitochondria. And so we think what we've identified is something that is leading to the accumulation of this dysfunctional mitochondria, where we think it's a two-part thing. First, you have that the mitophagy kinetics are just slow all on their own that could contribute to the buildup of these dysfunctional organelles. And second, you have the expression of these disease-linked mutations that we know affects mitochondrial health, but also decreases the efficiency of this mitophagy pathway. So we think it's the combination of these two factors that are leading to neurodegeneration, where what we see over time is that you get a buildup of these dysfunctional mitochondria. This leads to increased ROS and mitochondrial damage. This ultimately causes loss of mitochondrial network health, and then eventually you find degeneration of the neuron. And so what we're really interested in is seeing if we can identify some type of druggable target there to where we can speed up the efficiency of this mitochondrial to turnover of these damaged organelles, and would that be sufficient to kind of delay the onset of this neurodegeneration? And so before I finish with my acknowledgements, I just want to put a plug for this June issue of Cell that's focused on Juneteenth in STEM, where there are multiple articles that have been written by Black scientists. And so here I'm highlighting a commentary that was written by 61 Black scientists. And this was a huge effort that was led by A.J. Hinton, Ishmael Abdus-Sabor, as well as many others, that is really focused on some of the barriers to creating equitable science. And so the cover art, which I think is just really gorgeous, was done by Anthony Adonis Lewis, who's the father of Sam Lewis at UC Berkeley. So I just want to encourage you all to listen, learn from, and support your Black colleagues and fellow Black scientists. And so with that, I will conclude and thank my lab for all the hard work that they've done. It's been really great to work with everybody and getting the lab up and running, as well as our funding sources. some of the work that I highlighted from my postdoc with Erica Holzbauer, and the TBK1 story was from work with a senior graduate student in the lab, Olivia Harding. Also, we're looking for postdocs, and if you guys want to check out the lab website or follow us on Twitter, we'd be excited for that. Again, thank you so much for the opportunity to be here, and I'm happy to take questions.
That was wonderful. Thank you. And lots of beautiful images, which is something we always like to see.
Absolutely.
And lots of amazing techniques too. I mean, the optogenetics as well. So, you know, you've thrown the whole kitchen sink at it. So essentially, if you could find a drug that would help essentially with the clearing of those damaged mitochondria, then the thought is that you would prevent so it's like a congestion in the neuron that actually then leads to increase in its degeneration. Are they working on drugs like that you're aware of?
So there are different points within the pathway that I would say that most of the time they're targeted much more at the lysosome. So there's lots of lysosomal storage disorders that are also associated with that. So when the paper came out, there were a couple of pharmaceutical companies that were really excited to see that it could be potentially tied to the lysosome. And so trying to think about ways with which we could target that. The more data that we get, the more I'm kind of moving and thinking it may be slightly upstream of the lysosome, and that it may be more of a fusion defect between the lysosome and the autophagosome. But we have a couple of candidates that we are looking at now to see if one of those are involved, and if so, then I think that would be a really exciting target to kind of treat.
Awesome. Are there any questions from the audience? I haven't seen any in the chat, but I see that Feather's encouraging people, if you want to and you don't feel comfortable taking yourself off mute and asking, please go ahead and do it in the chat. We'll give people a few minutes, but yes, lovely work. And so what will be next steps for you with this work?
Yeah, so the next steps are really trying to characterize some of those targets or the potential proteins that we think are involved. Right now, we're trying to really understand, particularly whether or not those proteins are activated by oxidative stress. Because we think that is going to be very important in leading to this, because we know that there's such dramatic changes in the levels of oxidative stress, especially with age. Another thing we're trying to understand is whether or not those are actually, it's a bad thing. We assume that, you know, taking so long to remove these mitochondria is going to be bad, but oftentimes I get pushback, but like, is it really bad, or is that just our assumption? And so we're kind of trying to revisit that a little bit to really understand, why would the neuron do this? Is it in fact something that's beneficial for it, or that could be helping? And so one of our leading ideas is that it is useful in the sense that it allows the neuron to preserve components to where if it goes in any type of stress situation, it has everything it needs to recycle, to break down, to make anything new. And so we've been kind of testing, what does the rate of this pathway look like under low stress, high stress, different types of stress, to see if we can see changes in the rate of it, to kind of get at that question a bit more.
But I mean, have they also not shown that things like in neurofibrillary tangles that are in Alzheimer's disease, there's a lot of this sort of debris and junk. So, you know, it does sort of suggest, okay, if it's not able to clear it, the cell then tries to figure out some other way of compartmentalizing it and taking care of it so that it doesn't get in the way. And that then forms the basis of these tangles potentially.
Yes, so that's another exciting avenue, is kind of trying to understand, is it something that's specific to just the turnover of these mitochondria, or if it's something that's a little bit more general, and this happens to the multiple, the turnover of multiple organelles or aggregated proteins.
We have a question from Richard. Do you want to take yourself off mute and ask?
Yes. Hi, Dr. Evans. Beautiful work. I was wondering if this is mostly a somal phenomena, or do you also see this like at the synapses or in the axons? Yeah.
That's a really great question. So half of my postdoc work was trying to understand the location, and it's kind of complicated, but to simply put it, we did not see these optoneurin positive mitochondria within the axon. We found most of them only within the soma, and slightly within the dendrites that were pretty close, I'd say, nanometers away from the soma. And what was interesting is that we saw optoneurin within the axon, so we don't think it's something where the protein just isn't there, and so that's why it's not happening. But we think it may be linked to the fact that there are multiple types of mitophagy, and we just see that our optoneurin one is preferentially in the soma, and that other mitophagy pathways may be working out there within the axon. But overall, we think that the soma may be more of a reservoir for preserving, for either making new, so mitochondrial biogenesis and shipping things out to where they need to go, but also bringing everything back and then helping to get rid of it and turn it back over.
Really cool. Thank you.
Mm-hmm. Any other questions? So Chantell, you mentioned you had some potential job openings coming up. So I'm not sure if you're aware of the page that we have on the BINA website where you can post a job on the website and then it can go onto the Canada Bioimaging website, the Africa Bioimaging. Oh no, Africa Bioimaging is just doing events, Latin America Bioimaging. So it spreads it amongst all of the various network organizations and hopefully gets you a broader Canada pool.
I will absolutely take advantage of that.
Okay, yeah, I just dropped that in the chat. And then if there's anybody in the audience that's looking for jobs, they're there too. So
awesome. Let me go and copy and paste that. Thank you so much.
Yeah. Oh, we have another question. Oh, no, that's our hand clapping. Oh, my goodness, we're already past the hour. So that was wonderful. Thank you. Excellent.
Yes, thank you guys so much for the opportunity to be here, and everyone have a good day.